Yeah, so it's a very difficult question to answer right now, and that's because in triple-negative breast cancer, where immunotherapy is approved, particularly in the neoadjuvant setting, where I think we have a lot more data, all of the trials that we have are either small phase clinical trials, or there's only really one or two larger randomized clinical trials for us to explore biomarkers. So this is a really important question because unfortunately these drugs, while they have very durable responses and help patients live longer and even more patients be cured of their breast cancer in the early setting, there's really not a great way of telling who yet will respond and who won't. And unfortunately, these drugs can have very serious toxicities that are very different from what we're used to. They can cause autoimmunity, which, autoimmune-like diseases, which can affect any organ. These can cause permanent disabilities in the patients. And so this is a very important question. Right now, we don't have a good way in the early setting of identifying which patients will respond and which won't. The three most popular biomarkers are PD-L1 expression, which we're now confirmed does not seem to work in the early phase or the early setting. We have tumor mutation burden, which if it's very high, which is a very small proportion of patients, might predict some patients, but most of our breast cancer patients are in this intermediate range or in a very low range. And those patients sometimes still benefit from immunotherapy. And so at least at this stage, we can't use tumor mutation burden either. Tumor infiltrating lymphocytes, which is a somewhat easy way of looking at a biopsy from a patient's tumor and saying whether there are immune cells present in the tumor, also seems to hold some promise, but it's going to be very tricky because the patients that have lots of tumor infiltrating lymphocytes also are the patients that get the most benefit from chemotherapy alone and therefore might not actually need immunotherapy. There are a few additional biomarkers that are being explored in clinical trials in that early setting. And those include things that I'm interested in, like antigen presentation in the tumor microenvironment, which is a prerequisite for immunotherapy to work. Also, there are some really interesting studies looking at dynamic changes, so things that would happen on a repeat biopsy in a patient's tumor after they've started therapy. These are things like T-cell receptor clonality changes. They're changes in the peripheral blood, systemic changes that reflect a widespread autoimmune reaction against the tumor. They also include things like a repeat biopsy that you might be able to see what's going on in the tumor cell. Is it upregulating PD-L1? Are there more tumor infiltrating lymphocytes invading into the tumor? There's also other things that might not necessarily be specific to immunotherapy, but things like MRI changes or PET scan changes that are being explored in a lot of clinical trials right now. Unfortunately, now it's really tricky because in that setting, all the patients are getting immunotherapy, so it's very difficult to tease out what might be biomarkers that are general prognostic markers or those that identify the patients that are actually benefiting from the chemotherapy part and not the immunotherapy part. In the metastatic setting for triple-negative breast cancer, it's fairly clear from large phase 3 randomized trials that PD-L1 expression, if done correctly, can identify a subpopulation that's enriched for patients that are most likely to benefit. This is a very small percentage of patients that qualify for this biomarker, and it's actually not clear why many of the patients that are PD-L1 positive don't respond to the therapy. So I think we can do better in that setting. One of the biggest challenges there is access by academic researchers to be able to explore some of these things outside of industry, right? So many of the clinical trials are owned by industry and we can't access those in order to explore biomarkers that might help the patients better.